DNA replication. DNA is replicated in the S stage of interphase. The DNA needs to be replicated for a copy of the DNA to be passed on to the daughter cells. In mitosis, the parental somatic cell is diploid, and the two daughter cells are also diploid. In meiosis, the parental gonad cell is diploid, whilst the four daughter cells are haploid. The process of DNA replication involves 5 main enzymes: DNA helicase, RNA primase, DNA polymerase III, DNA polymerase I, DNA ligase. DNA is double-stranded. The template strand runs 3' to 5', whilst the coding strand runs 5' to 3'. DNA helicase separates the two strands by breaking the hydrogen bonds between the complementary base pairs. The place where the DNA strands are being separated is called the replication fork. The two strands are prevented from reforming the double helix by single-stranded binding proteins. Next, RNA primase inserts an RNA primer at the 3' end on the template strand and on the exposed 3' nucleotide of the coding strand. The DNA primer is an oligonucleotide synthesized by the RNA primase using RNA nucleotides. This is needed as the next enzyme, DNA polymerase III, also known as DNA polymerase delta, can only align nucleotides onto an existing nucleotide, which is on the RNA primer. DNA polymerase III reads the existing DNA strand from 3' to 5', laying down complementary bases running from 5' to 3'. DNA polymerase III gets the free nucleotides from the nucleoplasm, which is like the cytoplasm of the nucleus. The complementary pairs are laid down according to Chargaff's rule. The strand that is complementary to the template strand is called the leading strand, as it is made continuously. The strand that is complementary to the coding strand is called the lagging strand, as it is made in sections called Okazaki fragments. Once both the leading and lagging strands have been completely synthesized by DNA polymerase III, the enzymes DNA polymerase I, also known as DNA polymerase alpha, comes along to replace the RNA primers made of RNA nucleotides with DNA nucleotides. Finally, the enzyme DNA ligase stitches the Okazaki fragment backbones together. DNA polymerase II, also known as DNA polymerase beta, proofreads the DNA to check for errors and repairs it if a mistake is made.